Hello everyone, thank you so much for inviting me to share my work today. So my name is Kirill, I'm Assistant Professor, Department of Computer Science, and today I would like to share with you my work and my presentation titled From Protein Domains to Cancer Subtypes, Harnessing the Power of Multiscale Approaches to Advanced Biology. So my major contribution lies in advancing multiscale biomedical research by developing and applying computational tools to analyze and classify large biomedical data spanning across molecular, cellular, and tissue levels. And today I would like to share with you some exciting stories related to each of these levels of my work. So first of all, at the molecular level, during my postdoctoral work at University of Texas Southwestern Medical Center, I've been involved in the developing and maintaining the ECO database. It's evolution classification of protein domains. So we are classifying protein domains. Protein domains are functional and evolutionary conserved unit, and we can say that building blocks of proteins, and proteins are actually building blocks of our life. And today this classification can help us to better understand the evolutionary relationship and functional relationship of these proteins. So as you might know, today we have amazing AI-based tools which can help us to, for example, AlphaFold, which can help us to predict protein structure at the very high accuracy, almost as an experimental structure. And today we have millions of these structures. However, for the majority of these proteins, the functional relevance is really not very good understood. And that's here where classification can help us, because the idea of this classification is similar proteins might conduct similar functions, but as you can imagine, in nature it's not always that such straightforward approach works. So this database so far is one of the most comprehensive right now, and it includes experimental structures as well as a majority of AlphaFold models. So we included 48 whole proteomes of model organisms there, and using this database as a instrument, I've been working on several health-related research questions, which I would like to share briefly. So first question is, project is related to Salmonella enterica. This is a pathogenetic bacteria which causes severe disease in humans, and identifying so I've collected all genomes available from this bacteria and retrieved all proteomic data from that, and we're using AlphaFold. We modeled all the proteins and classifying them. So the feature of these bacteria is that they also include so-called Salmonella pathogenic islands. These are clusters of protein-coding genes which are involved in pathogenicity of this bacteria. So what actually does it mean? So these clusters of protein-coding genes, they encode this type of very complex protein machinery, which actually helps to inject the bacterial material into the host cells. And we identified more than 3,000 novel potentially pathogenic islands in these bacteria, which potentially can contribute to the living process of this bacteria and might be the drug targets for future approaches. Another project which I would like to share is a so-called pan-cancer structurome. So there is a genome, proteome, and we will look at the structurome. It's all domains related to 21 major cancer types and over and under expressed protein coding genes. We also used AlphaFold and all available experimental structures, and I looked at the distribution of all architectures for each particular cancer type of the all 21 types. And interestingly, I identified that overrepresented proteins in these all 21 cancer types were proteins exclusively beta architecture. These proteins are such as immunoglobulins, interleukins. This type of proteins involved in inflammation processes which often happen together with cancer genesis. And on the other hand, underrepresented proteins were exclusively alpha helical. So such proteins involved in homeostasis processes and transmembrane transport. So that kind of makes sense that cancer cells trying to reduce the representation of these proteins. And the most recent project which I've been working on related to drug discovery. So as we know, the majority of protein function related to interaction with the small organic molecules. And today, about 72% of all FDA approved drugs are actually small molecules. And however, the knowledge about which particular protein domain is binding, is coordinating particular drug is really missing from all actually available resources. So I've developed so-called drug domain database, which I started with human proteins, which are all major known drug targets, and for the small molecule drugs, I studied for all known experimental structures from protein data bank, and for the proteins without experimental structures, I use AlphaFold to model these structures, and AlphaFill approach to transplant miscellaneous ligands from experimental structures, and all this data reported in the drug domain database. Then I interested in the so-called post-translational modification. So that's a covalent binding of different chemical groups, covalent attachment of different groups to various amino acids of the proteins, and there are a large variety of them. So these are just some of them, like acetylation, phosphorylation. So actually the question is, what happened if post-translational modification occurs not far from binding site? Sometimes it's a really huge group. Would it be disruptive for the binding of a small molecule? Can it disrupt the functioning of the protein? So I've looked at the all known sites of post-translational modification and identified about 6,000 of small molecule binding associated PTMs, which locates around 10 angstroms around the binding site. And I used major AI-based tools which could model structures with post-translational modifications, and overall modeled about 14,000 PTM modified models also available in my database. And actually it turned out to be not very good studied question because of really lack of the experimental data at the structural level. And one of the interesting cases which I want to share is a phosphorylation of tyrosine of NADP cytochrome reductase. It's a very important enzyme. And as you can see here, so this is example of experimental structure of unmodified with the binding molecule. This is the AlphaFold model with the unmodified, and finally PTM modified model. And as you can see, the phosphorylation disrupts binding of this small molecule, and the size of the binding pocket is reduced significantly. So the interesting thing about this, that this type of phosphorylation was identified for this protein in 2 cancer types, and their functional relevance was unclear. And so my data show that it's actually might disrupt the functioning of this protein. So it kind of makes sense that it was found in the cancer, in 2 cancer types. And so right now I've updated my database to the version 2.0, and it includes all known experimental structures with the small molecules, so more organic or inorganic molecules. It includes AlphaFold models for human drugs targets without experimental structures, and extensive PTM-related structural data, which is one of the most, which actually the first database includes this PTM data at the large scale. So right now let's move on the cellular and tissue levels. So at these levels I've been working with the identification of subtypes of seminoma. It's the most common type of testicular germ cell tumors. So using available transcriptomic data and using computational approaches, I've identified two novel subtypes which we identified before, and we analyzed them using computational tools and identify significant molecular difference between these two subtypes. Moreover, subtype 2, we think that subtype 2 is a more aggressive type, and it's related actually by molecular features. It's closer to another testicular germ cell tumor type, which is more aggressive and require additional treatment approach, different treatment approach. We also looked at so-called microenvironment of these cancer tissues. So microenvironment is actually all cells which are around cancer cells, and the majority of them are immune-related cells. And we found that in these immune-related cells, we identified markers of so-called senescence. It's actually dormancy, we can say so. And we think that that might be the reason for failure of immunotherapy clinical trials for testicular germ cell tumors, because of the dormancy of immune cells which are targeting by this immunotherapy. And finally, these subtypes which are identified, they cannot be detected by pathologists just looking at the histopathological slides. So I've developed deep learning decision-making tool which actually can identify with some accuracy these subtypes, just taken as input as histopathological slides of these two subtypes. Now, what are my plans to? I plan to continue expansion of my drug domain database. My goal is to capture the entire universe of protein ligand interaction, which includes small molecules, polysaccharides, antibodies, nucleic acids, and so on. I plan to continue my work on the post-translational modification because it's really interesting and it's really not very well studied at the structural level. These types of modification are really crucial for our body and for protein functioning. For example, glycosylation is a very important hallmark of cancer, and glycans are used by, as you can see here, is SARS-CoV spike protein, which is covered by these glycans. They cover themselves with glycans to protect themselves from the immune system of the host cell. So that's very important mechanism to understand what's going on there. And finally, using my database, I plan to try to identify novel protein targets for drug repurposing. So it's drug repurposing. It's when we using the same drug to target different protein with a similar binding site, and also of target interaction based on this knowledge of evolutionary classification of protein domains. And I think that these resources also be very useful educational resource as well. Finally, I would like to share just one more direction which I've been working on with my friends and collaborators from University of Florida, which is one of my prospective direction as well. So they're working on so-called CRISPR-Cas systems. CRISPR-Cas systems are part of immune system of bacteria, and we are using these systems to modify genes actually in different ways. And the recent research shows that there are a large variety of these immune systems in bacteria, and the different types we can use as a different approaches to actually modify genes. And it's very interesting, useful, and in this work we showed this about the so-called Schlafens protein, and we show their antiphage, phase, and it's one of the most potential direction where we can find some more immune-related proteins in bacterial systems and apply them as gene-modifying objects. And finally, my teaching-related goal in the nearest maybe year or so, I plan to develop special topic course about structural bioinformatics, because it's very hot topic right now. It's very rapidly developing field, as you might heard about, that big tech companies like Google, DeepMind, and Meta, they already entered this field. They develop AlphaFold and SM-Fold, so-called. So, and the AlphaFold team got the Nobel Prize in chemistry last year, together with David Baker from University of Washington. So I believe that would be really interesting and useful for everyone who interested to enter this field. So finally, thank you so much for your attention, and I really appreciate support for all my collaborators from all over and from UCF, College of Engineering, Computer Science, and Computer Science Department. Thank you so much for your attention.